is Shauna Spiller. I am the soils biochemist at Quorum Laboratories, and I'm going to take just a few minutes to show you around your lab. This area is our basic prep area. We have everything that we need to process the soil samples, pH meter, shaker table, water bath. We have stir tables, we have weigh stations, analytical weigh stations. So everything that we do here is a prep area. Moving over to this area, this is our UV spectrometer. We use this to colorimetrically measure certain elements that are in the soil. For example, phosphorus, calcium, magnesium. We can colorimetrically measure those using the spectrometer. This machine is the TOC analyzer. This tells us it's a water extractable carbon. It will tell us the organic carbon, the inorganic carbon, and the total carbon that is water extractable. We can also use it under other extraction conditions, but for our purposes, we just use water right now. We do have an auto sampler on this unit, so we can load it up and let it run. It is an older unit, but it does the job. It gives us the information that we need. This piece of equipment is a gas chromatographer. What we're going to be using this for is to measure phosphorus levels as well as nitrates and ammonium that's in the soil solution. If you look inside, I'll give you an idea of how this works. The coil there, it's a capillary tubing. It's actually tubing. So the sample will actually travel through the capillary tubing. Depending on its mass, it can tell us which compound it is. We can also use this machine to measure all other compounds. You just need to know how much they weigh. So it's a really valuable piece of equipment. We are lucky enough to also have a hydrogen generator, which this machine requires hydrogen gas, and in order to be as safe as possible, we actually have a self-contained machine that will produce hydrogen for us rather than have to have an outside tank on this unit. So we have a safety hood that we use for noxious fumes, things that we don't want floating around in the lab. So we use this quite readily. We also have the ability to use vacuum and hook up gas to that so that we can have a flame inside the hood. Over here we have a centrifuge. Open it up so you can take a look. This centrifuge actually goes to 8,000 Gs, so it's a very, very powerful piece of equipment. And inside we load our tubes in here. We also have the ability to change these out and change the tube sizes. So we use this every single day. One of our pieces of equipment we use a lot. This is a vacuum dryer. Basically it will freeze dry samples so we can store them for long-term use. We haven't had a chance to operate this yet, but it's going to be, when we start getting a lot more samples, it's going to be good to have something like this because it really preserves the sample in its best form. We also have multiple refrigeration units that we store chemicals, samples, anything that needs to be refrigerated in the lab, so we have plenty of room for those things. We have another unit over there. Over on this side of the lab, this is the mass spectrometer. It takes up a lot of space. This is an ICP RQ mass spec, and it runs on the basis of a quadrupole, pole, which is 4 magnetically charged poles that are located vertically inside the unit. And when the sample passes through those poles, they actually switch on and off charge, and based on the speed at which the sample travels through, the element of the sample, we can determine not only the element, but the isotopes of the element. So some elements like calcium, lead, they have lots and lots of isotopes. So we're able to determine those. We have a 240 sample auto sampler unit, which is really nice. This machine is very fast. It can process 240 samples in approximately 8 hours. So we're very lucky to have this. It makes things very smooth and easy inside the lab. There is a lot that goes into this, and we're very happy to have it hooked up. We spent quite a bit of time preparing the lab and getting ready to get this piece of equipment in. So this is something we're going to be using a lot of. All right. go down the hall. In here. Okay. All right, this is our metagenomics lab. This is considered a clean area, so we don't let a lot of people come in and out of this lab because we don't want contamination issues, obviously. But inside here, we have a PCR machine. This is a PCR station, and what this does is it amplifies the DNA that we're able to extract from the samples. In addition to amplifying DNA, we can also amplify RNA if we need to or want to in the future. This unit is our Ion Chef and our S5 unit. What these do is this actually is a hands-free, clean, contamination-free prep area, and it's all robotic. If you want to show David, I just turned it on and the light will come on. But inside there's actually a robot, and all we have to do is load up all of the reagents, the pipette tips, and our samples and the chip, and it will automatically prepare the chip for us. This runs for about 8 hours, and so we can start it at night and come in the next morning, and it has a chip held for us ready to load into the S5. The S5 is a very powerful piece of equipment. This is a sequencer, and it is a semiconductor sequencer, which basically means that it functions in the same way that maybe a pH meter would function, and it measures the distribution of hydrogen ions over the course of the sequence to determine what the sequence is. So this is where we do most of our work with the sequencing. This information that's downloaded from this computer into our program actually gives us the sequence information as well as all kinds of other information about the sample. And there's all kinds of quality checks along the way, so we aren't wasting reagents or chips or other consumables. So there's just kind of an inside look at the Ion Chef. It actually does have a UV light, so it is all clean, so we don't have to worry about any kind of contamination on your chip. Over here, this is our Allergent bioanalyzer, and the purpose of this piece of equipment is to quantify DNA, and it actually can quantify your number of base pairs that you have. So in our work, we prefer to have anywhere from 200 to 400 base pairs in order to load our chip. So this machine can actually quantify and tell us how much of our DNA is within that 200 to 400 base pair range, so that we don't waste our samples or other consumables in the Ion Chef system. So we have another way to measure DNA. This is kind of a quick and dirty. This is an Invitrogen Qubit, and this is a fluorescent technology. So the DNA is fluorescently tagged, and we can also tag RNA, proteins, anything else that we want to tag. Stick the tube in there, and it will automatically give us a readout of how much that we have in the system. So. Alright, so this is our prep area that we do all of our genomic prep, DNA extractions. We have a real-time PCR system, which is very similar to the other PCR system in that it amplifies our DNA samples. But the nice thing about this one is we can actually quantify while we're doing it real time. So instead of, spending 6 hours on a PCR cycle, if we can see that our DNA is not progressing the way that we like, we can stop and try to figure out what's going wrong. So this is a really powerful piece of equipment. We also use a spectrometer to measure DNA levels in our sample. Just quantify the DNA. It's very simple. You put a drop of DNA on there, solution, and it'll tell you exactly how much is in the sample. We have the ability to do electrophoresis. We have 2 of these electrophoresis systems, which is basically a gel, an agarose gel that we can run out our samples on if we want to do any kind of expression work in the future. These come in really handy. This is a biosafety hood. This is actually certified for, I think, a bio level 2, but this can be used to do any kind of DNA work that you need to be extremely sterile for. We also have the ability to work with certain pathogenic substances that other labs may not be able to work with. So that's the purpose of this hood. We also have, of course, some centrifuges, other pieces of instrumentation that we need to use. This is our culture area. We have our culture hood. We prepare all of our liquid cultures in here and also our agarose cultures. Everything that we have in here is clean, just like the other lab, because we want to prevent cross-contamination. With soil samples, we typically do liquid culture. So we do have an autoclave over there that I'll show you that we autoclave all of our samples. Over here we have an area where we can do any kind of microscope work that we need to do. We have two pretty powerful microscopes. One is an actual stereoscope. And we actually have this hooked up to the computer system so we can image this, the information from the microscope directly onto the computer. We have another spectrometer. We have, I think, 4 or 5 of these in the lab, but they are very handy. This is an incubation unit. This actually incubates with carbon dioxide. It's a CO2 incubator, which is good for certain samples, and it's an option. So this is just a cabinet that we can adjust the temperature on to incubate different samples that we need to. We have shaker tables, UV lights. We even have our own water purifiers. Actually, we'll make ultra pure water for the lab so that we don't have to buy it constantly. That can tend to be an expense over time. The shaker tables, the 2 that we have, are used for incubating the samples. So as you can see, we have quite a bit of room to do a lot of work and do a lot of samples at once. This is our general biology area. Kind of do just about a little bit of everything in here. We store our chemicals in here. We have a sonicator, which comes in handy with certain experiments we need to sonicate. We have another spectrometer, lots of these in the lab. pH meters, we process a lot of the soil samples in here when they first come in, measure, weigh, quantify. Of course, sink areas, more centrifuges. We have another powerful centrifuge over there that we use just for biology samples. So we do have those separated. We don't want to mix up the biological with the chemical. We also have 2 autoclave units, which are for sterilization. When we are getting our matrix ready for culture. So that's what those are used for. And then this is just another general prep area that we utilize, as well as a hood for chemicals. We have lots of refrigeration units to store our chemicals and to store our reagents for our DNA work. So we have just about everything that we need in the lab. This is an orbital shaker. You can actually put water in here. Sometimes it requires an auto bath for culturing samples, and we can culture the samples at a specific temperature. That's the point of a water bath. So, it's a heavy door. So I just wanted to kind of give you a general overview of our lab. We actually do have everything that we need. The only thing that we might need along the way is chemicals, but we haven't had any issues thus far. We look forward to doing a lot more work in the lab. We have a lot of capabilities here, and I hope that we're able to do great things together. Okay, well, it was nice to talk to you, Mr. Lewis. Have a great day.